Okay folks, welcome to National 4-5 Genetic Engineering topic. So National 4 Key Area 3 and 8 are going to be covered, cell biology and the therapeutic use of cells. So insulin or other protein production via genetic engineering is what the National 4s need to know, and other examples may include stem cell technology, which we've done in a previous unit, or using cells to grow artificial organs. Now, if you've got time, you could have a wee look at these two documentaries. So, genetic engineering is a term used to describe the transfer of genes from one organism to another. Now, we know about genes. We know that in the nucleus you find chromosomes, and they're made of DNA, and a length of a chromosome is called a gene. What we're looking at here is cutting a gene out of one organism and transferring it to another. It's a technique of modern biotechnology. So, a wee bit of background. Diabetes is a disease caused by a defective gene. And this means that the body does not produce sufficient insulin. Now, what does that mean? Well, remember the genes have the instructions for making proteins. Now, insulin's a protein. So what that means is the genes got the wrong instructions. So insulin, the protein insulin, has the wrong order of amino acids and doesn't function properly. Now, why is this a problem? Well, insulin is required to control your blood sugar levels. So people that don't make correct insulin usually had insulin injections from cows and pigs, but this caused allergic reactions in some human beings because we're not pigs or cows and we can have allergies to that. So a human gene has since been inserted into a bacteria cell that produces human insulin, and that's what we're going to look at today. So bacteria. Genetic engineering involves the use of bacteria. Bacteria are useful because they multiply very quickly. Now, do you remember the very first unit we did, which was cell biology, when we looked at the structure of a bacteria cell? We said that bacteria are very primitive and they don't have a nucleus. Do you remember what they have instead? They have a plasmid, and we're going to look at the plasmid. We're going to use that today. So let's recap. Chromosomes, they're found in the nucleus of all cells. You've seen this slide before, we've used it quite a few times already. And we know that chromosomes are made of DNA. And we should know that DNA is made up of bases. And we should know that it's the order of the bases that gives us instructions for making proteins. And we know that a gene is a section of DNA that has instructions for making a protein and gives us a characteristic, for example, our hair colour, eye colour, etc. We should also know that our genome is all of the organism's genes. Now, what we're looking at in this diagram is we can see the two strands coiled together. We can see that's DNA, so that's a part of a chromosome. And what we're doing is we're cutting out a gene. So in genetic engineering, we're taking out a gene from one organism, which would be the human chromosome, and what we're going to do is insert it into the DNA from a different species. And what you're looking at here, the ring, is the plasmid from bacteria. So we're going to take out a gene from a human being and insert it into the plasmid of a bacteria cell. Now, the bacteria plasmid is instead of its nucleus. So it makes sense that the bacteria plasmid would be made of DNA. So if our chromosomes are made of DNA and our genes are made of DNA, well, the plasmid in a bacteria is made of DNA. So we can cut out the insulin gene from human chromosome and insert it into a bacteria plasmid. So we can see the bacteria cell there with the ring of DNA. We take that ring of DNA out, we cut it, we insert a gene, for example for insulin, and we put it back into the bacteria cell. Now we know that bacteria cells will multiply if they're cultured properly, if they've got the right nutrients to grow, and what we basically have is factories for making insulin, because the DNA has the code, the instructions for making the protein insulin. So the bacteria cells will make insulin protein because they all have the instructions now. So here we have our bacteria cell, and here underneath we have the human gene, which has been cut from a human chromosome. Can you see the plasmid being removed and cut at the top, and the gene being inserted into the bacteria plasmid? And then we put that altered plasmid back into the bacteria cell, and we culture the bacteria cell to make them multiply. We give them the right temperature, the right nutrients so that they multiply. And because every bacteria cell now has the instructions for making insulin, the bacteria cells will make insulin. Do you remember the structures required for protein synthesis? Ribosomes. Now you'll know from the first unit that bacteria cells have ribosomes. So the bacteria cells now have the instructions for making human insulin protein, and they also have the ribosomes. So bacteria is commonly the organism that a gene is placed into. This is because bacteria are simple organisms and reproduce very quickly, every 30 minutes. So a bacteria cell has a large circular chromosome, but it also has a plasmid made of DNA. So here we have the human chromosome with our desired gene in red. We're going to cut the gene out with special enzymes. So the gene is removed from a human chromosome. We're now going to remove the bacteria plasmid from the bacteria cell and cut it. And we're going to put the gene into the plasmid. Then we're going to put the altered plasmid with the human gene back into a bacteria cell. And we can see the altered plasmid there. The bacteria cell will make the protein that the new gene codes for. Remember, genes are instructions. So all we've given the bacteria is a set of instructions for making human insulin, and that will happen in the bacteria ribosomes. So the bacteria cell multiplies, and each bacteria cell will make human insulin, which can be purified for human use. Purification just means separating human insulin from all the other bacteria proteins that the bacteria cells will be producing. So a summary. Human chromosome, we cut out the gene of interest using an enzyme. We remove a plasmid from a bacteria cell and we cut it. We insert the gene of interest into the bacterial plasmid and we pop the plasmid back into a bacteria cell. The plasmids are going to duplicate, and we'll also culture the bacteria to make many copies. Each bacteria cell will have a copy of the instructions for making human insulin. Insulin is then mass produced. So let's just have a wee recap. How does the bacteria cell produce the insulin from the inserted gene? So if that's a DNA plasmid, basically we have the instructions for making human insulin in the bacteria cell. So the bacteria cell will make a copy of messenger RNA to take a complementary copy of the insulin gene and take it to the bacteria ribosome. So the messenger RNA will contain the instructions for making human insulin. We read the bases and put the amino acids into the correct order at the ribosome to make insulin. Remember, proteins are made of amino acids, but they have to be put in the correct order. But we have the instructions for making human insulin. We call the new bacteria transgenic. Advantages of genetic engineering. Completely different species can be combined, so we can transfer genes from any species into bacteria. The protein is easy to separate from the other proteins, which is purify. Alterations take place in only one generation, so it's fast. and large quantities of the protein can be produced. So there are disadvantages of genetic engineering. And the first one would be you can't make complex proteins in bacteria cells because they are quite primitive. So that restricts what proteins we can actually make. Now, so far we've looked at transferring genes from humans into bacteria plasmids, but there are other ways of transferring genes from one organism to another. And in this example, we're using a gene gun, which is mechanical injection of genes using a gun. So instead of bullets, you have genes that have been taken from an organism. And when you fire the genes into another organism, you're transferring genes from one organism into another one. But obviously, you're going to cause some damage to the organism that is receiving the new genes. Similarly, viruses can be used to carry a gene from one organism into another. But a virus can cause harm to the organism receiving the new genes because viruses can be dangerous. Another advantage is that if genetically engineered organisms, which are obviously altered in some way, they've got a new gene, a foreign gene in them, what could they do to the environment? Could they harm the environment? Could we create super weeds, for example, if we've changed a plant? And there's ethical concerns of putting genes from one organism into another organism. People feel it's too far, there's too much change, and it is quite expensive. So other controversial techniques include farming, which is the production of drugs, pharmaceuticals, medicines using genetically altered organisms. So that means that you're producing drugs, but in order to produce the drugs, you have inserted a gene from one organism into another. And obviously there is risk there. Any altered organism might affect the environment. So just to summarise, if you were asked about an advantage or disadvantage, here's the examples you could use. Advantages, you could say it's fast, you produce an unlimited quantity of protein, and it's production of human products, it's not from animals, so it reduces the risk of allergies. The disadvantages are its ethical and moral implications. It is expensive, but you could also say, again, do you remember the bacteria cells? That there was a limit to what proteins they could produce. They can't produce complex proteins. So the genetic engineering essay, what would that look like? So what we would say is a gene is identified and cut from a human chromosome. And then you have a bacteria and you remove the plasmid and cut the plasmid. And then you insert the human gene into the plasmid. So the altered plasmid with the human instructions is inserted back into your bacteria cell. Your bacteria cell is cultured so that it makes many, many copies, and your protein is purified, and that just means your protein is extracted, separated from all the other proteins that are being made in the bacteria cell. Why is bacteria used in scientific processes? It divides rapidly. What is genetic engineering? The transfer of genes from one organism to another organism. So what do we call an organism with a gene from another species? Transgenic. Why is genetic engineering seen as controversial? Transgenic organisms could affect the environment. So when you put a foreign gene into a new organism, for example a plant, and it's in the wild, it could affect other organisms in the environment. What is X? The plasmids. Now what is this whole technique called? Genetic engineering. Now let's have a look at exam style practice for National 5. The structure labelled X is plasmid. The diagram below shows a stage in the genetic engineering of a bacterium to allow it to produce human insulin. Which stage of the genetic engineering process is represented in the diagram? So we have an open plasmid. Good. Human insulin gene is being inserted into the plasmid. The diagram below represents some of the stages of genetic engineering which are used to produce medicines such as insulin for human use. Which one shows a bacteria cell? Remember, we're looking for a cell with a plasmid in it. Well done. E. Where's the insulin gene? So we're looking for a chromosome and a gene on it. Well done. C. And the plasmid would be D. Describe the next stage needed to produce insulin for use as a medicine. So we can see our insulin gene has been cut out of the human chromosome. The plasmid has been removed from bacteria and cut, and the human gene has been inserted into the plasmid, and the plasmid has been inserted into the bacteria cell. What would you need to do with that bacteria cell next? You would need to culture it so that the bacteria cell multiplied. So you need to culture it to multiply your bacteria to make many copies of insulin. Name another human hormone produced by genetic engineering. So we could have growth hormone, insulin, so any hormone that could be used to help human diseases. The following steps are involved in the process of genetic engineering. What is the correct sequence of these steps? So, use of an enzyme to cut out the gene from a human chromosome. Then use of an enzyme to cut a bacteria plasmid. Insert the gene into the plasmid and insert the plasmid back into the bacteria cell. Identify S and T. So S is the gene and T is the plasmid. Again, identify Y and X. So X is a plasmid and Y is a gene. The following statements describe processes involved in genetic engineering. You have to put them in the correct order. Okay, W, V, Z, Y and X. So the required gene is identified and cut from the human chromosome. The plasmid is removed from the bacteria cell. The gene is sealed into the plasmids. The altered plasmid is put back into the bacterial host cell. And what you want is your new bacteria cell to multiply, making many copies of your protein. So name structure X taken from a human cell. That is your human chromosome. What is the chemical? From what chemical is section Y made? Section Y is identified and cut out from the chromosome. Y is a gene, and genes and chromosomes and plasmids are all made of DNA. What type of single-celled organism is being used in this procedure? Bacteria. The diagram below represents stages in the process of recombinant DNA technology, genetic engineering. So, genetic engineering involves insertion of a gene from one species into another, into which structure from the bacteria cell is a human gene inserted. The plasmid. The diagram below shows stages in the production of a desired product by genetic engineering. Which line in the table identifies correctly the structures labelled above? X is and Y is. So X is the plasmid and Y is your gene taken from the human chromosome. And remember, human chromosomes, genes and plasmids are all made of DNA. Which line in the table below identifies correctly one advantage and one disadvantage of genetic engineering to make desired products? So an advantage is fast production, a disadvantage is it's costly. Now you can have a look at other disadvantages. Possible release of genetic engineered bacteria into the environment because you're changing organisms. You don't know what effect it's going to have on the environment. And the other advantage would be increased range of products. Which stage in the production of human insulin by genetic engineering is represented in the diagram below? We can see the human gene being inserted into the plasmids. State one advantage of genetic engineering. Increased range of products, fast production rate. Okay folks, genetic engineering can be used to transfer human genes to bacteria artificially. So we've got to fill in these boxes here. So let's start with chromosome extracted from human cell and required gene located. What's the next thing? You locate the gene. What's the next thing you do with the gene? You cut the gene from the human chromosome. Now, if you look to the right, bacteria cell carrying plasmid identified. What do we do with the plasmids? We remove it and then cut it. So what would stage 3 be? We've got a cut bit of plasmid and we've got a gene cut out of a chromosome. We'd be hoping to seal the gene into the plasmid. And then what are we going to do with the plasmid? Put it back into the bacteria cell. And then we can see at the bottom, grow large quantities of the bacteria by culturing it properly and then purifying it, which means separating your protein from all the other proteins. Now I'm going to stop there. You can continue with the questions. The answers will follow. And just practice this PowerPoint before you go into class.